Hi everyone and welcome to this lecture. In this lecture we're going to focus on the beginning portion of chapter 4 and our topic is psychopharmacology. Psychopharmacology is just simply the study of the effect of drugs on your nervous system and on behavior. So first let's define what a drug actually is. It is defined as an exogenous chemical, meaning that it is a chemical that is not produced by you within your body. It is something that is produced outside of your body, so it's something that has been manufactured, and it is something that is not necessary for normal cellular function. So meaning that you could survive without the drug, or that a typical person could survive without the drug. And additionally, it is something that, when taken in relatively small or low doses, alters the functions of certain cells within the body. The effects can either change in, can produce some kind of a change in either a physiological process, so an actual bodily function, or it can produce a change in behavior. You're going to hear this term throughout your reading in this chapter, this sites of action. What this means is basically at what point the drug molecules interact with molecules in your body's cells. So where on a cell in your body, one of your own cells, does the drug interact? Now let's focus on the drugs a bit for a second. So pharmacokinetics, this is focusing again on the drugs, and we focus on how drugs are absorbed within the body. This will be heavily dependent upon how the drug is administered, and we will go through in this portion of our lecture for this chapter how drugs may be administered. Also, how they are distributed throughout your body. They can be through your blood and your circulatory system, including your central nervous system. How they are metabolized or broken down by your body. This typically involves some change into an inactive form of the drug. So the drug eventually becomes inactive in your system, most likely by enzymes produced in your liver. And then how the drug is secreted. So how does your body finally, once it has been distributed throughout your body and then metabolized by your body, how does your body then get rid of it? Typically, it is through your kidneys. So typically, it is metabolized. Your kidneys get the end part of it, and then it will be secreted through your urine. Now let's take a look at routes of administration. So how can we administer a drug? The first is IV or intravenous injection. So with this, the injection is into the vein. The drug will be absorbed and distributed almost immediately when given this way. It may not be time for the drug to be counteracted though if too much is used. So for example, if a dosage is not prescribed the right way, or if this is a self-administration issue here, if too much is administered, it is absorbed so quickly into the body that it may not be possible to counteract the effect of an overdose. There is also IP or intraperitoneal injection. This is when we inject into the abdomen. This is typically done with non-human subjects. Again, this is an injection into the abdominal wall here. Again, a lot of times this is done in laboratory environments, lab animals. We can also do IM, which is an intramuscular injection. So this would be into the buttock or into the thigh area, into the upper arm. If you've ever had a flu shot, you've had an IM injection. This way, when we administer in this particular way, the drug is absorbed through your capillary system in the muscle. So the absorption won't be quite as quick with IM as it is with intravenous. We can also do a subcutaneous injection. So this is going to be the injection right under the skin. So this isn't as deep. So here we can kind of see the angles of injection here on the right-hand side of our screen. So intramuscular, as you can see, goes in at a 90-degree angle, so it's going to go pretty deep into that muscle there. Subcutaneous is going here at a 45-degree angle, not as deep. And then, of course, we can do oral administration. We would do this for tablets and liquids. This is most common, of course, for folks at home taking therapeutics. There is also sublingual administration. This is when something is administered under the tongue and it will be absorbed that way. Some drugs are destroyed by your digestive system. So, for example, insulin, someone who is diabetic who takes insulin, that type of drug will be destroyed if it is taken orally. Your digestive tract and the acids and things like that in your digestive system will destroy them or break them down and make them basically inactive. So sometimes something like a sublingual administration would be what is needed. And when we give something in this way, it absorbs into the bloodstream by the capillary system in the mucous membrane of your mouth. This is commonly done for migraine treatments, also nitroglycerin tablets for folks that have heart issues. A lot of times they will be given what is called nitro quick or nitroglycerin tabs. They are taken under the tongue. They tend to become more active quicker than if they take them as an oral administration. So if you were to swallow something, it's going to take longer for it to become active because it has to be slowly broken down. Whereas if it's taken sublingually, the effect of the drug will happen much quicker, which for someone who has a heart condition, if they need nitroglycerin very quickly, then they would want to take something sublingual so the effects of that drug can happen more quickly. And one more additional thing, this also obviously tends to be easier on the stomach, this administration, because it isn't going through your digestive system. It's not going into your stomach. We can also administer drugs via inhalation. This is going to be when somebody actually inhales the drug and it actually reaches their lungs. This is common in asthma treatment, for example. The effects of this are very rapid. We can also do topical administration or administration on the skin. This is done for a variety of different hormone treatments and also, for example, nicotine patches as well. There is also something called insufflation. This is different than inhalation. When a drug is given via inhalation, it reaches the lungs. With insufflation, it does not reach the lungs. It actually reaches the nasal mucosa and enters the brain that way. This can be also referred to as snorting. This is done with recreational drugs, but this is also the way of administering drugs for anesthesia as well. And the effects of insufflation are very rapid, almost immediate. There are occasions where we may want to focus on a drug's effect within the brain. If that is the case, we might do one of two administrations. The first is intracerebral administration. This is what is done when we want to observe the effect of a drug on a particular brain area or a direct administration. So a direct brain injection is done into a specific region of the brain, and then it is absorbed into the brain tissue, and we can observe the effects of the drug on that particular area of the brain. However, sometimes we may be more interested in more widespread absorption into the brain as a whole. If this is the case, then we would do intracerebroventricular administration, or ICV. This is where we are interested in injecting into the brain, but there being more widespread distribution of the drug. So we want it to spread throughout the brain. Now, if you remember from our previous discussions in earlier lectures, there is the blood-brain barrier. This kind of gets around the blood-brain barrier, this direct injection. I would like to emphasize that this is rarely done, somewhat rarely done. When it is done, it is typically done to deliver antibiotics directly into the brain. This might be done for someone with terminal cancer, and that would be really the only case that I can think of that they would use this particular method in humans. These methods in general, these two that we're looking at right here, are more commonly done in non-human subjects. Now that we're done with routes of administration, I wanted to show you this quick graph here on the right of this screen. So this is looking at cocaine concentration and its effectiveness, or how concentrated it is in the body over time in minutes. And this is going to vary dependent upon the route of administration. So here the black dotted line is intravenous. So this is if it's injected into the vein. Here we have smoked That is the orange line. Then we have orally administered, the blue line, and then intranasal, here the purple line. So what we can see is that the concentration of cocaine in the body is much higher initially than in the other methods, but it also declines much more quickly than the other methods. Whereas these tend to be a bit more gradual, slightly more gradual, but then decline with time, as does the intravenous injection. So now let's chat about drug effectiveness. With the exception of the direct brain injection that we looked at in the previous slide, all of the administration methods that we talked about so far, they only vary in the rate in which the drug actually reaches the circulatory system. So how quickly does it actually reach your circulatory system and get circulated throughout your system? There is something to consider, which is lipid solubility or fat soluble. Is the substance soluble in fat or not? Again, this is the ability for fat-based molecules to enter the cell membranes. So the cells within your body, if something is fat-based, is it able to enter your cells or not? Lipid-soluble molecules or fat-soluble molecules pass into the cell linings of the capillaries in your system and into the central nervous system. For example, heroin and morphine are examples of this. They are soluble, so they can pass through the linings of your capillaries and into your central nervous system. These are going to be equally effective, these two, heroin and morphine. Keep in mind here, one is more of a street drug, one is more of a prescriptive drug, morphine. Both are equally effective, but the difference here is that heroin is more lipid soluble. So it's going to more efficiently pass into the capillaries and into the central nervous system. And that is why for some individuals, heroin, unfortunately, is more preferred. When it gets to the point of breaking down drugs within your system and then finally getting rid of them or them leaving your body, drugs are commonly metabolized through enzymes in your liver. Some of these enzymes are also found in your blood and in your brain as well. And when it's finally time for them to leave your body altogether and be excreted, they've been broken down and they're ready to leave your system, they are primarily excreted through your kidneys. Some enzymes in your body, once they interact with drug molecules, when they go to actually metabolize them, they metabolize the drug molecules and actually form something even more effective. So these drugs tend to be very long lasting. An example of this would be Librium. This is a drug that's commonly given for anxiety. A dose of this drug can last for up to 24 hours because once it enters your system, the enzymes in your body that begin to metabolize the drug transform the drug molecules of Librium into something that's more long lasting. And now we'll take a look at effectiveness, so how effective a drug actually is. This is commonly mapped by using what's called a dose response curve. So given the effect of, given how high of a dose you're taking, how effective is the drug? What this curve is meant to do is to map the effectiveness as a function of how much drug was actually administered. So what we know is that different doses are needed depending upon the organism's size. As you notice, a lot of times when you go to doctors' offices, they take your weight and your height. This is very important because this will, if they need to prescribe you something, this will help them determine how much of a drug to give you. A larger individual is likely going to need a larger dose of a drug than will a much smaller individual. So that is very important to take into account when prescribing a drug dosage. What we call something having a maximum effect. Basically, this is when the effect of the drug will plateau, and it will plateau at a certain dose. And whatever dose that is the dose that's going to give the maximum effect. So we can see that here on this curve that we're looking at here on the right-hand side of the screen, this dose response curve. Right here we have this plateau or this flattening off of the function. And this is when we're getting a high effect of the drug. And again, this is plotted as a function of drug dose. So here we're getting more toward the high range of the drug dosage. And at this point, at some point, the effect of the drug kind of hits a ceiling. So it can only be so effective. So that's really the dose where we want to hit at. We don't want to go any higher than that because it's not going to produce any better or as stronger of an effect. Something else we have to note is that drugs do have multiple effects. They have the intended effect, but they also have other effects as well. For example, opiates. They're meant to produce analgesia or kind of numbness. However, what they can also do is depress neuron activity in your medulla. Your medulla, if you all remember from previous lectures, is a part of the oldest part of the brain, and it really helps with control of heart rate and respiration or breathing. So what this tells us is that while an opiate can be really good at producing numbness, for example, or analgesia, it can also actually depress your respiration such that you would stop breathing and your heart would stop beating. So these are things that we have to take into account when we are prescribing drugs. They do have the intended effect, but they also have other effects as well. Now, something we also want to focus on when we're looking at that dose response curve is also the effectiveness of the drug. And the effectiveness, of course, is the drug producing the desired effect, but also we need to think about what is called the margin of safety. As we just discussed in our previous slide, drugs have the intended effect, but also unintended effects as well. And these factor in to what is referred to as a therapeutic index. Now, what this does is basically tell us what the safe dosage would be for a drug. So this would be for both humans and non-humans. So what we want to obtain if we're, for example, testing a drug, we would want to find a dose of that drug that produces the desired effects for at least 50% of the population we're testing, but also the dose that produces toxic effects in less than 50% of the population we're testing. So this index, this therapeutic index, is the ratio between these two things. What dose produces desired effects in most, but also produces toxic effects in the least. So a higher index is desirable. So a higher index number is really what we want. So we want to find a dosage of a drug that produces way more desired effects than toxic effects. So we want the dosage that is high here, but also at that same dosage produces few toxic effects here. That's going to give us a higher ratio number, which is what we want. That is more desirable. What that translates to us is that this drug at this dosage is safer. So the higher the therapeutic index number is for a drug at a given dosage, the safer it is. So for example, we'll talk about barbiturates here in a second, but barbiturates are a class of drugs that are very low in therapeutic index. Some are as low as 2 or 3. To contrast that, benzodiazepines, which are more commonly used these days than barbiturates, for example, Valium, are much more safe. These have therapeutic index numbers of over 100. So benzodiazepines are kind of used more so these days in place of barbiturates because they have a much higher therapeutic index and they are thought of as being more safer because of that. As you can see here, the barbiturate numbers being as low as 2 or 3, that means that there is a much higher likelihood that you will wind up with toxic effects or an overdose situation because the margin for error, or in this case, if we're looking at this curve here, the margin of safety is very, very narrow with this class of drugs. I mentioned barbiturates in the last slide. I wanted to focus on them for a few more minutes here in this lecture. Barbiturates are typically given as anti-anxiety meds or to have some sort of sedative effect or be even a sleep inducer. And again, these are kind of an older class of drugs. They're not really used as much. And again, as I mentioned in the last slide, that is because they are thought of as being less safe. They interact with your body as an agonist for GABA. Now, GABA is a neurotransmitter. We will learn about GABA and other neurotransmitters when we do part 2 of this chapter next week. But GABA is a substance in your body, a naturally occurring substance in your body, a neurotransmitter, and it has a overall calming effect on your system. So we also say that an inhibitory neurotransmitter. So what you need to know for now is that GABA inhibits certain activities, so relaxes muscles, for example, and things like that. Barbiturates are going to be agonistic of GABA, making it more active, making the effects of GABA more active, so relax your muscles more, for example. Barbiturates are very, very strong. The effects of some of them may last up to 2 complete days. Again, these are less commonly used now because they have a very low therapeutic index, making it much more likely that someone could overdose on these type of drugs. These are just some examples are barbiturates. We have phenobarbital and sodium thiopental. That's more commonly referred to as truth serum. So again, these are drugs that are not given as commonly now as they were years ago. Now, I also want to distinguish between opiate and opioid. A lot of times folks are using these terms interchangeably. They are not the same. Opiates are drugs that are derived from an opium poppy plant. So for example, heroin, as I mentioned earlier in this lecture, as well as cocaine and codeine. Opioids are either natural or synthetic, and they bind to opioid receptors in your brain. So opioids can be natural, but they can also be synthetic or man-made. Some synthetic examples would be hydrocodone or Vicodin and OxyContin. Now, things to keep in mind here at the difference between opiate and opioid. All opiates are opioids, but not all opioids are opiates. Natural isn't necessarily better or safer. If you notice the synthetics here, hydrocodone, oxycodone, fentanyl, these are drugs that are synthetic, man-made, that we use by prescription and also use in surgeries and things like that. These can be used in a way that is much safer than natural occurring substances like cocaine, heroin, things like that. Now let's look at affinity. Affinity just refers to how well molecules join together. That's very important when you're thinking about drugs and how they affect the body. So we're thinking about how drugs, in particular the molecules of a drug, bind to its receptor site on cells in your body. So if a drug has high affinity, it means it's very effective at low doses, meaning that it is very good at binding to receptor sites of your molecules, even when given at low doses. A low affinity drug means that we need a lot more of the drug to be administered to have the same effect. So it doesn't bind quite as well, so we need to administer more of it. Ideally, what we would want is a drug that would be high affinity for therapeutic sites of action, so the sites that we want it to bind to on cells within your body, but have very low affinity for toxic sites of action. So again, we want it to be very high in action in the way that we want it to act, but very low in terms of binding to areas that are going to produce a toxic effect. So let's look at what happens when we administer drugs repeatedly. What we can have is obviously tolerance developing. So tolerance is basically your body's attempt to compensate for the effects of the drug. So your body is naturally going to produce some kind of compensatory reaction to a drug. So it's going to try to counteract somewhat what the drug is doing. This is again referred to as being the act of tolerance. So again, your body is going to try to attempt at staying at some kind of optimal set point. So when you take a drug, your body is going to produce some kind of reaction in response to that drug. When we have withdrawal symptoms, what's happening is that the drug is no longer being administered and it's stopped suddenly, but your body hasn't stopped producing that compensatory tolerance response. So your body is still producing tolerant responses, but the drug isn't being administered anymore. So you're feeling your body's reaction to repeated administrations of that drug now that the drug is being removed. Withdrawal symptoms can occur by, of course, decreasing in binding or decrease in receptors. So this can happen when you take a drug for a very prolonged period of time. Overall, that drug's not as effective anymore. You may have to take more of that drug to get the same effect. And this is happening because over time, with a repeated usage of a drug, its ability to bind to receptor sites in cells in your body is getting lower and lower. It's not able to bind as well. This can be because the receptor sites are changing in shape. It could also be that the receptors are kind of dying off. So the receptor sites, the places on the molecules in your body that can bind with the drug, are either changing in shape or they're dying off. We can also have instances of physical dependence. So this is what's going to occur when withdrawal symptoms are experienced by an individual. Other things that can happen with repeated administration are things like sensitization. So this is where we actually have greater effects of repeated dosages. So with repeated dosages, your body is producing greater effects, or the drug is giving greater effects to your body. This is much less common than tolerance. Tolerance is much more common. Sensitization may occur to some effects, but we also may have tolerance of others. So the same drug could produce sensitization in some ways, but produce tolerance in other ways. For example, cocaine. Cocaine produces more movement disorders and seizures, so more sensitization, but it also has euphoric effects, which is why most people take it, but those effects may show tolerance. So while you are using more cocaine, you may be getting less euphoric effects from it because you're getting tolerant to that effect, but you may also becoming more sensitized to the movement issues that can happen with repeated cocaine usage. So you may be getting less of the desired euphoric effects, but more of the movement issues and seizures that can come along with repeated cocaine usage. Now we discuss placebo effect. You guys have probably heard of the term placebo at some point in your education. Placebo is just an inactive substance. It can be thought of as being like a sugar pill or something, but basically it's anything that is meant to mimic a drug but really isn't doing anything. It's incorrect to say that a placebo has no effect because in many cases, if someone believes that they're receiving a substance that is going to do something, they're likely to report feeling effects of it even though it's not doing anything. And physiologically, it is possible to have the body actually mimic the effects of a drug if the individual thinks that they are receiving the drug. So we can have a physiological effect, but also a psychological effect of placebo. This can take place because of motivational issues or expectations. So if you expect to feel something from administration, you may say that you actually do feel it, even though what you received was an inactive substance. Most studies that are done, most drug studies are required to use a placebo or what is also commonly called a control group to compare results to. The FDA requires most to use a placebo group. Last, we'll look at sites of action of a drug. So these are those places on a cell where the drug is actually going to bind and have its effect. So drugs that affect synaptic transmission fall into two groups. Synaptic transmission, meaning how substances are being transferred across a synapse or where two neurons meet. They fall into two groups, and I kind of touched on this earlier when I was talking about barbiturates. There are some groups that are agonists, like barbiturates are. So agonists facilitate that transmission across the gap between neurons. So it's going to increase activity. There are also those called antagonists. They do the opposite. They block or inhibit synaptic effects. So they block transmission of neurotransmitters, making activity decrease, for example. Okay, folks, at this point, we are done for this portion of this chapter for this week. So we've went through routes of administration, so different ways in which we can administer drugs. We've also talked about the safety of drugs, focusing on the therapeutic index of drugs, and also we have begun to talk about how drugs bind to receptor sites on the molecules in your body. And given how they do that, what effect they may have on your functioning. Next week, we will continue with this chapter and we will start talking about the different types of neurotransmitters that are in your body and what they do for us, and also how a variety of different drugs interact with those neurotransmitters and their effectiveness. So that is all for now. Please make sure to read through this portion of this chapter carefully and have a wonderful day.